Hi everybody, this is an AP Biology video about mitosis. So let's start talking about why do cells divide. So here we have a cell and it has a red chromosome and a blue chromosome. And before it can divide, each of those chromosomes need to make a copy of each other. That's called DNA replication, and it happens before mitosis. And then here, this cell is in the stages of mitosis because you can see the chromosomes lined up at the middle and you can see these spindle fibers. And then if you look after it, you see that the chromosomes have separated and moved to opposite poles. And you end up with two cells that are identical to your parent cell. So this is a great example of what happens during mitosis. And a little bit about what happens before mitosis. So what's the goal of cells dividing? You divide so that you can make new cells. The cells that are made are going to be just like the parent cell. So it's really helpful when you're talking about asexual reproduction, like budding or fission. Those are both forms of asexual reproduction that just make new cells identical to the parent. And when you think about your body, all the growth that happens from when you're a baby till now is all due to cells dividing and reproducing. And that is what mitosis is all about, helping you grow. So before we get too into mitosis, we have to talk about DNA a little bit. So DNA, when it's not condensed, it's called chromatin. And it looks like a net. You can't really tell anything apart. If I was going to draw a cell and I was going to show you chromatin, I would draw the nucleus and then I would just make it look all stringy in here. You can't really tell one chromosome from another, but all of that is chromatin. All right. Now, chromosomes. If I could see individual strands, each strand of DNA is one chromosome. And each body cell that you have, so if you think about cells in your arm and cells in your stomach and cells in your cheek, each of those cells are called body cells or somatic cells. They're all diploid. And the way we write diploid is we write 2n. N is the number of chromosomes that you have, and 2n is your diploid number. So for humans, we have a total of 46 chromosomes. That is our diploid number. There's 23 different types of chromosomes, and 2 times that is 46. All right, moving on. The way that you end up with 2 of each chromosome is that you're getting 1 from mom in the egg, and you're getting 1 from dad in the sperm. So both of those chromosomes contain genes for the same traits. So let's draw an example down here. I'm going to draw, here's 1 chromosome. I'm going to make it blue. That is a chromosome from dad. And then I'm going to make this one red. That's a chromosome from mom. If they are the same chromosome, so let's say that these are both they're both chromosome number 2. All right. They'll probably be right around the same size. They have different genes on them. So from here to here is one gene, and here to here is another gene, and another gene, and another, and another, and it keeps going. And there can be over 100, up to 1,000 genes on one chromosome. And over here, mom has the exact same genes. Now, she does not have to have the same form of a gene. So let me explain what that means. Right here, let's say that this is a gene for if you can roll your tongue or not. So dad has the dominant form of the tongue rolling gene. That means he can roll his tongue. And over here, the same spot, mom has a little a. That means she has the recessive form of the gene. That means she's unable to roll her tongue. So 2 chromosomes, they both carry the gene for tongue rolling, but mom has the form of the gene that cannot roll, and dad has the form of the gene that can. So you have 46 chromosomes. They are numbered 1 to 23. Numbers 1 to 22 are called your autosomes, and pair number 23 are your sex chromosomes. If you end up with an XX, you are a female. And if you end up with an XY, you are a male. All right, so let's move on. Now that we've talked a little bit about DNA. Most people think chromosomes look like this. So most people are like, wait a minute, why didn't you draw an X? An X is actually a replicated chromosome. So once the DNA copies itself, it looks like an X. And there's some parts of this we have to talk about. First of all, this part where they connect right here, that is called a centrosome or a centromere. Sorry, it's really hard to write with the mouse and the pen, but you get the idea. All right, that is a centromere or a centrosome. Means the same thing. All right, whether I put centromere or centrosome here. All right, now this little section right here and right here are attachment points. They're special proteins, and that protein is called a kinetochore. A kinetochore is where the spindle fibers attach to a centromere. So here's my replicated chromosome, and this is where it's going to attach, right here and right here. That's the kinetochore. All right, so now we've gone through what the DNA is all about, and now we're ready. We can talk about mitosis. So mitosis is part of a cell's life. You call a cell's life a cell cycle. And I'm sure you guys remember this word, interphase. So interphase is where a cell spends about 90% of its life. So the majority of time when you look at a cell, like if I said, oh, go look at these cells under the microscope, you'd be like, oh, they're so boring. That's because 90% of them are going to be in interphase. The only ones that aren't in interphase are the ones that are dividing. So there's three parts of interphase. The first one is called G1, and the G stands for growth. So G1, the first stage of growth. Then the next part of interphase is called S. S stands for synthesis, and what's being synthesized is DNA. So remember how I just showed you a chromosome that was replicated? All of that has to happen. Your chromosomes have to go from being strands to being X's because they've copied themselves. So that's what happens during S. Then the last part of interphase is called G2. And I want you to think about this for a second. You're not just replicating the nucleus, you have to replicate everything. If you're about to make 2 cells out of 1 cell, you're going to need double the mitochondria, double the lysosomes, double the ER, double the ribosomes. You got to make more of everything. So the cell really needs to grow a lot. So G2, you got to really think about that's going to be replicating all of your organelles. I liked this visual. You can certainly grab this off Google or draw it into your notebook, but it starts right here, G1. So this is the beginning of a cell's life. It's growing, and then S is replicating its chromosomes, and then G2 growing anymore. Now these are talking about checkpoints in these sections, and G0 is talking about when a cell stops growing. So each of those are going to be something that is in another video. The checkpoints allow cells to only divide when necessary. So it makes a lot of sense that we're going to have periods where our cells are arrested and not dividing. If you think about you for a minute, most of your cells are done growing, done dividing. You might still be getting a little bit taller, or you might be developing more cells, but many of your cells, like for example your liver, those cells aren't dividing anymore. Your muscles, even when you get more muscle, the number of cells isn't increasing, just the proteins are. So most of your cells are going to be in G0. All right, then over here, this is mitosis, so this is where the stages of dividing is going to happen, and then cytokinesis is where the cytoplasm splits and you end up with 2 cells. All right, so let's get into these stages of mitosis. Again, this should be a little bit of a review from Regents Bio for you. We got to talk about what's happening. So in mitosis, the cell is dividing. It's producing cells that are just like the parent. There are 4 phases. All right, so I'm just going to open this up so you see all of it. The first phase is called prophase. So pro always means early. So the first phase. In this phase, chromosomes condense and become very visible. So if you look at my drawing, we now have these puffy X's, and the puffy X's are showing that the chromosomes are very visible. They've condensed and thickened. The nuclear membrane dissolves. That's shown in white right here. So in order to get the chromosomes to opposite sides, you got to break apart your nuclear membrane. And then the centrioles are here and here, and they have moved to the opposite poles, and these yellow spindle fibers have formed. And they're reaching out to grab onto that kinetochore region of the chromosome. All right, so that's what we got happening in prophase. The next phase is called metaphase, and metaphase means they line up at the middle. So all 4 of these chromosomes have lined up across the center. The centriole spindle fibers have attached to the kinetochore of all of the centrosomes. So that's what metaphase is. Very easy to recognize, but very, very short in terms of how long cells are at that phase. They're going to spend far longer in prophase than they are in metaphase, because a lot more has to happen in prophase than metaphase. All right, next phase is anaphase, and this is where a cell starts to split apart, and it splits the sister chromatids. So you call each replicate chromosome a sister chromatid. They have now separated, and they're being pulled as the spindle fibers shorten. Now, if you have an animal cell, it tends to look like this. This pinching in is called a cleavage furrow. Whereas if you had a plant cell has that boxy look to it, so it forms what's called a cell plate across the middle. All right, and then last we have telophase. So telophase is the final division. You got to start reforming, there it is in white, reforming your cell membrane. Your chromosomes start to decondense, which means they're going to thin out and get stringy and look like chromatin again. This drawing tried to show you a little bit of both. The cell plate looks sort of like that, the yellow part, and then the pinching in is the cleavage furrow. So it's showing you a little bit of each in this diagram, but you should definitely know that cleavage furrow is for an animal cell and cell plate is for a plant cell. And then this word, cytokinesis, means that we can't just talk about the nuclear division. You have to remember that all of the cytoplasm is going to split and go to each side. That's going to happen concurrently. So cytokinesis, splitting of the cytoplasm, happening at the same time. All right, that is the end of our mitosis video.